So, very often I've heard you speaking about the concept of the human microbiome symbiosis. And I've heard you talking about how maybe perhaps we're less than 50% human. So,
Yep.
could you dive into this and start off for the listener explaining this concept of the
Sure, with
human
pleasure.
microbiome symbiosis?
Yeah, so we often say we're microbial, and this is already very important. Meaning by that we are a mix of human cells and microbial cells. And in numbers, this is 50 trillion bacteria with which we are interacting on a constant basis. And recent calculations actually that were made by colleagues from Israel told us that we are roughly a little bit less than 50 trillion bacteria, but also 50 trillion human cells, if you account for all the hematopoietic stem cells, among others, that were not counted before. So for a long time, we were saying we were 10 human and 90% microbial. I think the recent figure tell us we are nearly 50-50. And what's important there is that we are not only host to bacteria. It's really a constant interaction. And our microbiota, our gut microbiota, probably will emphasize that, is providing essential protective functions. Just to roughly summarize that, it's contributing to digestion of the food that we do not digest because we're not equipped in terms of enzymes to digest them. So they essentially pass through the intestine all the way to the large intestine, where we have the high density, high diversity microbiota. And there, obviously, they serve as food, as carbon and energy source for our microbes. And so that's the contribution number one, let's say. Contribution number two is that our microbiota exerts this protective functionality. It prevents from a proliferation of bacteria that we ingest, for example. Every piece of cheese we eat is 100 million microbes. Every piece of cheese.
Wow.
And not all nice and friendly, but in fact they have no chance to establish, none of them, because of our microbiota and its barrier effect, we sometimes call it. Number three, they keep our immune system educated and ready to react all the time. So this is a very important interaction with our natural defenses, and it keeps our natural defenses ready to act to manage any disruption, any distortion, any threat, basically. And it can be a microbial threat, but it can also be a threat of our human cells when they deviate from normality, when we have a beginning of a tumor, for example. Then it's handled by our immune system, and normally it's eliminated. And the education of our immune system by our gut microbiota is important in that respect also. It's the same with interaction with a response to vaccination, for example. The efficacy of vaccination is somewhat modulated by our microbiota. And then the last protective functionality or functionality that I may mention is actually interaction with each and every cell, tissue, organ of our body. So we talk of axis. So we talk of the gut-liver axis, the gut-lung axis, or the gut-heart, or sometimes the gut-brain axis.
So one of the questions that might be popping up for the listener is, that all sounds incredible. How do we know that the microbiome contributes these functions? What are the experiments that we've done to get an understanding of these areas?
Yeah, so this brings us back a few decades ago, actually, when people did a lot of work to try and breed animals under completely sterile conditions. And it became possible to obtain baby mice, for example, under sterile conditions. So that's what we call germ-free animals. And then quite rapidly it became possible to compare germ-free animals with their conventional counterparts that were living outside with the microbes. So the germ-free ones are raised in sterile containers, boxes, or big chambers that you access with gloves, basically. So, and everything and we use to host them is sterile. So sterile food, sterile water, sterile beddings, and sterile cages. And so they can live a whole life without microbes, actually rodents can. And then you can inoculate them with the normal microbiota of the same species, or you can inoculate them with a microbiota from humans, for example, or other species. And the comparison really was very clear in terms of documenting some of the functionalities we mentioned. For example, I mentioned protection against proliferation of environmental microbes. If you raise an animal under sterile conditions and then you expose it to other bacteria, and not necessarily severe pathogenic bacteria that produce factors, but just basic bacteria, sometimes they can actually be contaminated and die from this exposure. So really having the normal gut microbiota is a strong protection against potential infection, even by harsh pathogens. And obviously the education of the immune system you can evidence by looking at the gut tissues that host the immune system, and you see the maturation of lymphoid tissues around the gut system that is really established when and only when there is exposure to microbes. Now, it's a research tool to have animals that are raised under sterile conditions. Humans are not raised under sterile conditions, obviously. So we are microbial from the moment of birth on, basically.
Yeah. So as soon as we come into the world, they colonize our body top to bottom, inside out. Maybe this is a good opportunity for you to talk about how does the microbiome then develop? And which years are important? What do we understand about how your start in life might influence your later health due to the microbiome?
The moment of birth is very special in that respect. It's a moment where we meet the microbial world for the first time. We are in an environment which is virtually sterile before that. And colonization is rather well known in the early stages. So we know that there is what we call succession. So the ecosystem, the gut environment is not strictly speaking aerobic. So there is some exposure to oxygen from the maternal blood that circulates in the baby's body. And so it's oxygenic in a way, or there is some oxidative stress, so to speak. And then the first colonizers are microbes that within the maternal vaginal microbiota, for example, or intestinal microbiota as well, are resistant to exposure to oxidative stress. They are facultative anaerobe, as we call them, and they prepare the ecosystem for colonization later on by strict anaerobic bacteria that are really the dominant ones in the gut microbes of every adult human. And so there is a succession that we know fairly well, and diversification takes a few years, actually. Initially, you have just a few species of microbes that are established, and they prepare space and leave space to others in dominance. And for quite some time, we thought that after the age of 3, everything was roughly at the level of diversity or richness as what we'd see in the adult. A recent paper told us that, or still at the age of 5, it's still in construction in terms of richness and diversity. So it takes years to establish. What we can say also is that in the early times of life, diet, in a way, feeding is important. So breastfeeding versus bottle feeding makes a big difference. And although things have improved in terms of bottle milk mimicking maternal milk, much better today than when I started working on that, still there is a faster, more rapid diversification with bottle milk than with maternal milk, which tends to promote bifidobacteria, for example, or lactic acid bacteria that feed on lactose from the maternal milk. And they're also controlled by oligosaccharides, very specific oligosaccharides that are present in maternal milk and that condition somehow the maintenance of a less diverse microbiota. And also what we can say is that the mode of birth will be very important. So you can be born by vaginal delivery or by cesarean section. And cesarean section, obviously taking the baby by an operation, does not allow exposure to maternal, vaginal, and intestinal microbes. So it somehow delays the diversification of the microbiota and also because there is no exposure to maternal microbes as efficiently as by vaginal delivery, then it's not only a delay, it takes much more time to diversify. It's even documented as associated with a smaller but higher risk of development of chronic conditions later in life.
Yeah.
What more can we say? There's a very nice recent publications in terms of epidemiology saying that we do expose our young ones early on in life to antibiotics much more today or in the recent times than obviously 50 years ago or more. And that has a very strong impact on the establishment of the microbiota, on its diversification. And it's also associated with higher risks of many chronic conditions that have been tested. And this is data from epidemiological studies that were undertaken comparing thousands of infants or individuals. So it's very robust in terms of epidemiology, let's say.